All right. Hello, everybody. Welcome back to the Planetary Sample Seminar this week. I am pleased to introduce Dr. Laura Rodriguez. She'll be giving our talk today. Dr. Laura Rodriguez is a staff scientist at the Lunar and Planetary Institute and an organic geochemist with a PhD in astrobiology and geosciences from Penn State. Her research interests are centered on elucidating how planetary environments drive the chemical evolution of organic matter to evaluate the potential for the origin of life and other worlds. To this end, Dr. Rodriguez performs laboratory studies of abiotic reactions and develops computational strategies to elucidate the organic content and samples analyzed by the planetary spacecraft or returned samples. Currently, she is also funded to build the Astrobiology Spectral database, a repository for abiotic organic spectra. Dr. Rodriguez is a participating scientist for the MSL Curiosity rover, a member of the MSR Campaign Science Group, and a recipient of the NASA Planetary Science Early Career Award. Dr. Rodriguez, I will let you take it away. Thanks so much for being here.
Hi, thank you for having me and for taking time out of your day to attend this talk. Just want to do a quick sound check.
We can hear you here.
Okay, great, fantastic. So today I'm going to be talking about how planetary science can benefit origin of life and some of the issues that we have with the origin of life. It's a very difficult and unconstrained problem because one, we only have one data point for where we think an origin of life event occurred, and that's here on Earth. And so it's really difficult from just one data point to be able to infer trends or constraints on what's needed for an origin of life event. In addition to that, we don't have any rocks from the time that we think the life emerged on Earth. So we're even at a loss with that one data point. And there's a lot of unknowns regarding the geochemistry of the early Earth. And so it's a really difficult problem to tackle. Laboratory work provides an excellent analog for how we think the planetary environments can facilitate chemical evolution. And we can test this using specific conditions, but we're also really constrained on what we can simulate in the laboratory because it can never really approach the true chemical complexity that we would expect on a planetary environment. And so because of that, it's really important to benchmark what we learn in the lab with samples either brought back or analyzed in situ on planetary missions. Unfortunately, origin of life is not really prioritized for planetary missions. It's not necessarily written into mission objectives. And so the reason for that is because planetary missions require science closure, and it can be really difficult to write an objective regarding origin of life that has that closure. How do you write it so that you can say you know for certain you've addressed whether a planetary body could have hosted an origin of life event, since there's so many things we don't understand about the origin of life even on Earth. And so because of that, planetary missions really tend to focus on the questions in astrobiology, such as, are worlds habitable? Because we know what it takes for a planetary body to be habitable, at least for Earth-like life, right? You need liquid water, you need bioessential elements like the carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, and you need a source of organic compounds and energy. And so there tends to be a lot of focus on that in planetary missions, as well as now there's a growing interest in having missions address the question, could there be life on other worlds, either in the past or currently? And I would say that it's a detriment to cut out origin of life from the research objectives of planetary missions because it's really key to being able to answer that second question in particular. And that's because prebiotic chemistry, abiotic chemistry can look very lifelike. It can look like the signatures of life. And so we need to have a good understanding of how complex abiotic chemistry can get in order to be confident in our assessment whether we found actual evidence of life or something that just looks lifelike. It also can help inform us as to what planetary bodies you may want to focus on or what in particular aspects of the world we want to look at because we think that those are most likely to have hosted an origin of life event. And so what this figure is demonstrating is just how complex we know abiotic chemistry can get. So everything here has been demonstrated in the literature to a point. And so, starting from the left-hand side, very simple organic molecules such as formaldehyde, cyanide, we know those exist in abiotic environments. We see them even in the interstellar medium. We know through lab work and through analysis of asteroids and meteorites that they can combine to form biologically relevant molecules, including nucleobases, such as the adenine shown here that stores your information in RNA and DNA, as well as sugars and other amino acids, for instance. And in the lab, we've seen that these compounds under certain conditions they can link together in what we call polymerize and form longer macromolecules, such as RNA-like molecules. Even fatty acids with a long hydrocarbon tail, you throw them in water, they spontaneously form these vesicles that look like cells. And it's been shown in the literature that these vesicles can absolutely incorporate floating nearby macromolecules, and that those macromolecules can then undergo reactions within those vesicles. This is all abiotic chemistry. And so if you saw something like that, if you brought like a microscope and you were looking at some samples and you saw that, you might be convinced that it's life, but it's really not. It's just complex abiotic chemistry. And so because of that, it's really important to have a good understanding, not only of how complex abiotic chemistry can get, but how does it look with instrumentation that we take to other planetary bodies, because we're going to be really limited with in situ life detection and what we can take. And so another point I want to bring up is, if you've heard of the ladder of life detection, one of the compounds that's been highlighted as a potential smoking gun for life is pigments. But we know that molecules of pigments can actually form abiotically. Now, I'm not saying that chlorophyll in particular spontaneously arises in the abiotic environment, but the core structure of chlorophyll can, which is the porphyrin ring shown here. And abiotic chemistry can show that it can actually incorporate metals as well. And so there are abiotic pigments and molecules that look like pigments, at least spectroscopically, that you could find in an abiotic environment. And if you're limited with, let's say, just Raman spectroscopy, you might think you found them, but they're actually just abiotic. And so it's really important to be able to study these more and understand how they can form abiotically. And so one way that we can start incorporating origin of life research into mission objectives is if we have some kind of framework, so that it's not just the question of, was an origin of life event possible, but we can start asking more pointed questions to test origin of life hypotheses. And so that's what this proposed framework is doing. So we know, starting from the left, there's the simplest organic chemistry you can get is forming simple volatile organic compounds, low molecular weight, like cyanide, formaldehyde, from simple gases. We know that then those can form to produce biologically relevant building blocks like amino acids and nucleobases. And under certain conditions, those can form larger macromolecules such as peptides and nucleotides, which are the precursors for proteins and nucleic acids, respectively. And if you really have organic chemistry go on a little bit too productively, you can start to form this tar, which is also referred to as insoluble organic matter, or IOM. Whether this IOM material is actually productive for abiotic chemistry is unknown. It's actually the largest component of organic compounds in carbonaceous chondrites and cometary material as well. And so it's really important to be able to study how that could interact with this environment and whether that represents a dead end potentially for abiotic chemistry. Of course, you could also recycle IOM material back into smaller biological building blocks. And so if you start putting planetary bodies on this framework, we could start asking more pointed questions, such as I have on B, Enceladus, with the biological building blocks. It's been proposed that for origin of life, you need some kind of land, because to form more complex molecules, you have to have what's called condensation reactions. Condensation reaction is just chemical reaction between two molecules. When you form a new bond, it produces water, and that's a condensation reaction. Unfortunately, if you try to do this in bulk water or bulk ocean, like in Enceladus, you have your products surrounding you, so the water can react with the formed larger compound and reform your reactant. And so it tends to favor those reactants rather than the compound, since you're surrounded by water. And so because of this, it might be difficult to produce larger macromolecules like RNA and DNA, cellular membranes, proteins, in ocean conditions, because all of these are actually formed through condensation reactions. And so it's been put forth by Damer and Deamer, for instance, that maybe ocean worlds are habitable but uninhabited because you don't have access to land, you don't have a way to really drive this chemistry forward. We'll say this hasn't been really well tested extensively in the lab because there are other potential mechanisms for having these reactions be favorable in water. But that's one hypothesis that's been put forward. And that's really interesting because one aspect, one thing that we can do with observables from missions is try to evaluate the likelihood that they stem from life. So Affolter et al. In 2021, they did a study looking at the hydrogen and methane ratios measured by Cassini of Enceladus's plume to try to evaluate the Bayesian probability that the ratio was due to life versus not life. And Bayesian probability is really interesting because it's been talked about in life detection circles as a way to evaluate in future life detection missions whether we have life or not. And so this really depends ultimately on, they talk about it in the paper, on knowing the probability for an origin of life event. And so ultimately, if you cannot have an origin of life event, that drives the probability to zero. So they did all this work, but it's really a function of the probability of an origin of life in which we don't know. And so if we could address, like, yeah, you need land, then that would help, evaluate whether observables like what was made by Cassini, whether those are actually due to life or not. And so going back to the framework, this is how we can incorporate origin of life hypotheses in future missions. So the next problem with origin of life research is it's actually really complex to analyze abiotic organic chemistry. It's very challenging to analyze. And that's because abiotic organic chemistry generates all these different products that aren't really relevant for life as we know it today. And so that's what this figure is demonstrating. So we have several different mass spectrum. So we have on the x-axis mass charge ratio, intensity on the y. And each of those peaks represents a different compound, maybe several different compounds if they have the same exact molecular mass. And it's just demonstrating that there's a ton of organic compounds. They're generated abiotically, such as through Miller-Urey chemistry. You just have atmosphere, water, lightning, tons of organic compounds. Extractions of organics from carbonaceous chondrites, tons of organic compounds we've seen as well. When Cassini flew through the plume of Enceladus, it didn't use a high-resolution mass spectrometer, but nonetheless, we were able to detect that there were probably a lot of different organic compounds there, and some of them may have been really high molecular weight. So probably some complex organic chemistry going on there. And even on Titan, where the temperatures are really, really, really cold, and you would think you might be hindered by the cold temperatures and the type of organic chemistry you can get, there's still a lot of organics that are generated through UV irradiation of the atmosphere. And that's been shown with simulations of Titan chemistry. And so because of that, we are limited in what we can report in any one study. You can't go and track down and fully characterize every single peak you'll have in a mass spectrum. So we tend to focus only on the biologically relevant compounds because of that. And unfortunately, that means a lot of the organic chemistry that's happening is ignored and not studied. Another problem with trying to go and, even if you really wanted to go and focus on these different compounds, since we don't have commercial standards, because commercial standards are very biased towards biology and drugs, it's really difficult to say anything with certainty about these compounds. You'd have to probably try to generate them synthetically, your own standards, which is very complex. And there's just no way you can do this for all of the organic compounds you're generating. So it really limits what we can say about abiotic chemistry outside of what's relevant to Earth-like life right now. And so in some, because of this, origin of life studies are really biased towards biologically relevant compounds. We tend to ignore everything else. It's unreported, it's unknown. And that really hinders our ability to evaluate how the environment can drive chemical evolution if we're ignoring most of what's being produced abiotically. And it's important to note that biologically relevant compounds are important for life today, but it might not have been as relevant for an origin of life event or the earliest forms of life. So now I want to talk about a case study kind of highlighting this with RNA precursors. So RNA is composed of nucleobases, sugars, and phosphate. The sugar in particular is ribose, and then the nucleobases we have here. And when you combine the nucleobases and ribose, they form a nucleoside. So that is a subunit of RNA. And that reaction itself is a condensation reaction, so it can be difficult to achieve in water. And previous studies have already established the nucleobases under even the most optimal conditions do not react with ribose to form this nucleoside, not like direct chemistry such as that, just adding the two. The pyrimidines, which are the one ring heterocycles to the right, those three, don't form it at all. They don't react with the sugar. The purines, the two rings, adenine and guanine there, do, but they're very low yield. And considering this is dry, with really high concentrations, it's not very promising that you would produce a lot of this in the abiotic environment. And so it's been thought that these types of molecules are actually unreactive because of these studies. And so here comes my PhD work. I did this with Michael Cahan from Boise State and Chris House from Penn State. And I was investigating the reactivity of nucleobases in a foremost reaction, which is the polymerization of formaldehyde, or the repeated addition of formaldehyde, and you form larger and larger sugars. So this is a leading theory for how you can get sugars on early Earth, and potentially also in asteroid parent bodies. And so the idea was we wanted to see if you could get nucleoside through this type of chemistry. And so the foremost reaction was performed under alkaline conditions, and we incubated it with the nucleobases between 5 to 80 C, up to 54 hours. We analyzed it with Orbitrap mass spectrometry, which is a high-resolution mass spectrometer, which means that for essentially every peak that we got, we could narrow down what the chemical formula was. So here's an example of binding a nucleobase with the foremost reaction. And what's highlighted here, the green peaks are peaks that are only found in the reaction mixture. So they are products. And then what I have highlighted are some examples of the chemical formula that we know and what tentative structures. So these structures are not confirmed, just to give you an idea of the chemistry that was happening with this nucleobase. And I'm able to infer that structure because since I now I'm starting with the binding nucleobase, I can subtract that chemical formula from the chemical formula of the product mass, and what's left over is the structure. And so one thing to note is that there are a lot of different peaks here. This is just one part of the spectrum. So it's actually quite reactive, unlike what we expected. We expected these one-ring nucleobases to not form anything of interest. But here we are seeing a lot of things of interest that actually look very nucleoside-like. And so the next step is to isolate that ions of interest and then fragment them further to get a mass fragmentation profile. And this can help us deduce what the structure is. And so the way that works is you isolate. So like what this red, green, pink blob here that I have is like let's say a molecule. You bombard it. For instance, you can bombard it with electrons or some gas. And labile functional groups will pop off. So for instance, the first one that pops off in this example is that red group, which let's say represents carboxylic acid. So that can pop off and you have a new peak that's detected. And that can further interact with, let's say, electrons, and the other groups can pop off. And so here's an example of butanone and its mass fragmentation profile. So for instance, you can chop off that first methyl group and you'll have a 57 fragment, whereas the whole molecule itself is 72. So that's the parent molecule. When you pop off the methyl group, you have 57. Or if you pop off the carboxylic, the carbonyl group with the methyl group, you'll have a fragment of 43. And so you can see that you can start correlating the different fragments to the structure of the molecule. And so this is one way that we can deduce what the chemistry is. So I talked about, we found the nucleobases are very reactive, but when we actually compare the mass fragmentation profiles with nucleoside standards, because we did find compounds that had the same chemical formula as the biological nucleosides, which was surprising, but we also knew there's no way that can be it because there was all these studies done way back in the 1970s showing that doesn't happen. And we confirmed by comparing the fragmentation profiles that it is not the same. But nonetheless, they're still very reactive. And so we had a follow-up study where we reacted the nucleobases with ribose, and we let the ribose degrade because sugars are very sensitive. So you heat them up long enough, they degrade very quickly. And then we analyzed the product from that, and we found we're still getting all of these really interesting nucleoside-like compounds. So it's not reacting with sugars, but with their degradation products. And what this is showing here, this figure, is the comparison of what was identified as a product, nucleoside-like. It has the same chemical formula as a nucleoside, but it's a different mass fragmentation profile, which is what we would expect. We don't expect this kind of chemistry to happen where you form the nucleoside. But even though it's not the same, it's quite distinct. It looks similar. So if you just had a high-resolution mass spectrometer, you might be like, oh, man, I formed a there's a nucleoside here, abiotically. It's so exciting. But it's not really. But nonetheless, it's still really interesting, and it could be useful potentially for origin of life chemistry. We just don't know. But they're still really interesting, and it shows that these compounds are still very reactive. And if you were just focused on the biological nucleobase, you wouldn't know that. But this approach was still very biased. I only found what I looked for. I only looked for certain masses based on chemistry that I thought could be happening. And so now I want to talk about how we can use computational strategies to facilitate organic characterization in an unbiased way. And this is really relevant because this requires high-resolution mass spectrometry. This is really relevant for Europa Clipper because it actually has a high-resolution mass spectrometer, the mass specs instrument. And so what we develop here could be useful for and applied using that instrument. And so I'm going to be talking about two different goals. One is developing scripts to pre-process mass spectrometry data, and the second is talking about machine learning. And so the first is to develop scripts that can identify, deconvolute, and characterize peaks, because you can actually do a lot of interesting chemistry just with that without using machine learning. And so one way to do this is by having a moving spectral window that goes through your whole mass spectrum autonomously in small pieces to get like little curves. And then what you can do is try to fit those curves to a function. In this case, I'm using a pseudo-voigt function, which is a combination of Gaussian and a Lorentzian function. So Gaussian is like that bell-shaped curve. Lorentzian is a little bit more narrow. And what this equation on the bottom is showing is the pseudo-voigt is a combination of those with the n and 1 minus n showing the proportion of Gaussian versus Lorentzian. So if you solve this equation, which has variables for full width hat max, master charge, central master charge peak, intensity, and that n, you can replicate the function here if you get good parameters for each of those. And so from optimizing that, we can then get the parameters for the peaks. And so first, to do this, you have to have a starting guess for each of these parameters as well as constraints, and we can get those using a combination of first and second derivatives. And then we actually optimize the function using a Levenberg-Marquardt optimizer. And one of the things that I've learned is that it's really important. Your starting guess is really, really important because depending on your variables, let's say this is a space representing the different combinations you can have for the starting guess. So let's say if you started here with your combination, you can get stuck in local minima that don't really, in other words, your fit function might not fit very well with the curve, but it's optimized. So it's really important to try to do a grid search of combinations to ensure that you're actually capturing the global maximum of really good fit. And so what this is highlighting is how the script, it works really well even with peaks in the noise, with really intense peaks, and it's really useful for deconvolving peaks, as you can see here. And this is just a real quick way to get the peaks in your sample. And then from there, once you have the accurate mass, you can start to solve for the chemical formula, especially if you have constraints like, so for my reactions, since I reacted it with thymine or with the nucleobases, and I'm only looking for peaks that are only in the products and not in the control reaction mixtures, I can say, I can put some constraints on the chemical formula. And the chemical formula is further constrained by the accurate masses of carbon, hydrogen, and other elements. And so what this is doing is it's using a linear optimizer called Pulp in Python to essentially combine different amounts of the elements to minimize the difference between the observed mass and the theoretical mass that's calculated. And then from there, when we have a good chemical formula, then we can subtract out the chemical formula of the nucleobase from that to get the chemical formula of the side chain. And then from there, based on that chemical formula, we can apply certain rules to classify the reaction as whether the side chain is a carbonyl or sugar-like or sugar alcohol-like. And so applying this to my reaction, so for instance, this is adenine with the formous reaction at ADC at 3 different time points. We can build histograms then of the types of structures that are being added to the nucleobase. And so right away you can see most of the compounds that are being formed have a carbonyl side chain. And looking at the different colors, for instance, you can see that this carbonyl chemistry happens even at T0, which means I had the nucleobase, I had the formless reaction, I mixed them, and then froze them at minus ADC, and only thaw them right before I analyze them. And even then, you quickly form these compounds. Another point to bring up is the sugar alcohols group, which is really interesting. So those are nucleoside-like. They don't have a ring like you have in nucleoside because it's a straight chain sugar alcohol. So you have an extra OH group, but they're still really interesting because they're still similar, and they are forming also very rapidly. Another thing you can do when you have this type of information is then start making plots of, for instance, a particular group. So I've grouped the sugar, deoxysugar, and sugar alcohol side chains. And now I'm plotting for each reaction the distribution by the number of carbons. And so you can see with all of the nucleobases, except 5-hydroxymethyl-uracil, which is sometimes substituted in RNA for uracil. All the others, though, the main side chain is 6 carbons long. So we tend to get these sugar-like 6 carbon long side chains, and they do form very rapidly, even at T0. So another step that you can take is trying to leverage machine learning. So we were able to get as far as we could in characterizing the structure without using, for instance, mass fragmentation profiles. But if you have that, then you can get a little bit more specific and more confident as to the compound that is being generated. And we can use, in particular, I'm using the data that's freely available from metabolomic databases that have a lot of mass fragmentation profiles of compounds, and training machine learning models using that to classify compounds based off their mass fragmentation profile. So, for instance, an example of classification would be saying it's an amino acid, or maybe we can get even more specific and say it's an alpha versus beta versus gamma amino acid. And so what I started with my initial model was using unsupervised learning first to learn associations between the MS peaks, or the mass fragmentation peaks, using an algorithm called Spectral. And this itself is based off of word2vec, which is how we can learn associations between words. So for instance, if you had like a book, the word2vec algorithm can see how often words co-occur to deduce relationships, so how similar words are to one another. And it represents these similarities using vectors. In this case, this figure, the vectors are represented by colored boxes. So king and man, for instance, have more in common than king and woman. And so you can see that the colored boxes for king and man are more similar than they are between king and woman. So taking that forward with Spectral, it's looking to see when masses from fragmentation profiles co-occur. And from there, saying whether they are similar. And so the vector that's produced from that, you can then compare the vector shape rather than the absolute vector magnitude. You want to compare the vector shape to see how similar they are to one another. And the idea behind comparing the shape versus the magnitude is if you compared the absolute, let's say the absolute peaks of a compound. So what this figure is showing at the top, let's say you had compound A with a little extra functional group, let's say a methyl, and you have a fragmentation profile, and even though A with a little methyl group is very similar to just A with no methyl group, the absolute peaks aren't going to be the same because there'll be a little bit of an offset because occasionally a fragment has a methyl group. And so if you're just comparing the absolute masses, then you might say they're actually not very similar because a lot of the masses don't overlap. And so because of that, you really want to be able to accommodate for that. And so comparing vector shape versus magnitude is one way to do that. And so then we use these features we get, and we can then put them in a supervised learning model to predict the chemical class. And the chemical class is based off of an organic taxonomy that each of these fragmentation profiles have in the database. And so these databases use classifier is what it's called. And classifier essentially, sorry, classifier provides this organic taxonomy for each of the entries. And then we can use the model, in this case, gradient boosting, to predict that taxonomy. And so that is what this is showing an example, a confusion matrix. It's one way to evaluate how well your model is performing. So we have the true label on the y-axis and the predicted label on the x. And so if it's a perfect prediction, you would expect a solid blue line, all ones on the diagonal. Anything falling off of that is a misclassification. Please note that this is not all of the classes that were tested. There were too many to show. You wouldn't be able to read it. So this is just an example of some of the classes that were tested. One thing you can take away right away is if you look at amino acids and derivatives, you'll see that it was for every amino acid and derivative that was it was perfectly classified as that. However, other compounds were misclassified as that. And so you can see that by looking at everything falling off of that one. There were a lot of compound classes that were misclassified. That's probably because the and derivatives is probably a very broad group and might actually be very challenging to accurately classify groups when they're very broad because they probably have a lot of overlapping functional groups. And so one way that that's highlighted is if you take those Spectral embeddings and we plot them using t-SNE, this is just showing how the data is dispersed. You can see here, for instance, in the red, the aminosaccharides, they start forming these distinct subgroups. And so you can imagine a model trying to classify all of this. It might have trouble versus if it was just having to classify these subgroups. And so it might be easier, I thought, to classify based on the subgroup, which is more specific than the larger parent group. And so I tested this with my favorite molecules, nitrogen heterocycles, which include the nucleobases. And sure enough, the model accuracy improved a little bit. So it's promising that you can use this type of model for predicting even more specific classes. So instead of just saying you have a nitrogen heterocycle, for instance, you could say with more confidence that you have a pyridine, such as D, the one nitrogen ring, or pyrimidine with two nitrogens, which is shown in E. So machine learning has a lot of promise. Unfortunately, you need a lot of data for machine learning. And planetary science in particular is relatively data poor compared to other fields. And that's because we're just limited on the number of samples that we have and what we can do in the laboratory. So here comes my potential proposed solution, which is the Astrobiology Spectral database, which I refer to as ASD. And this is supported through a NASA Planetary Science Early Career Award. And the idea behind this is that if you want to really start leveraging machine learning for life dissection and characterizing organic compounds, we really need a good database that's well curated, specifically for abiotic organic chemistry. We just don't have anything like that exists right now. All the databases that I'm using right now are biologically focused. It's metabolomic databases. And so we need a place where people can store their mass spectra or other spectra of abiotic compounds, because, as I mentioned before, there's only so much you can do in a publication, and you don't want all of that other data that you don't talk about to get lost. It could be useful for other researchers in the future. And so because of that, I wanted to create this repository. And my idea being that, like how missions preserve 75% of their return samples, this database can preserve data for future scientists. So maybe we learn that, hey, this is really an interesting compound that's diagnostic of a particular chemical process. Let's see if it's, in these other spectral databases, or in these other spectral data sets. So that's one way that it could be really important. In addition, I mentioned that a lot of the abiotic compounds we just don't have commercial standards for. It's really hard to characterize these things. And so having a database will enable us to be able to generate machine learning models that are actually tailored for characterizing abiotic organic chemistry. And ideally, with this type of work and this type of resource, it will equip us with what we need to go from seeing the trees, from just inventorying biologically relevant compounds, to being able to see the forest, being able to understand how the environment can drive chemical evolution of organic matter. And so this database aims to be a fair and functional database. We aim to have it focused on storing mass spec and NMR data to begin with, because these are the two gold standard methods for characterizing organic compounds, but we are open to eventually taking in other spectral data sets such as Raman or UV-Vis. My co-Is on this work are from the Open Data Repository, Barbara Lafuente and Nate Stone, and I also have a science committee composed of Lori Barge, Andrew Rios, and Ram Krishnamurthy, Shrimi, to discuss, to provide input into the development of this database. And so I wanted to talk a little bit about the objectives, what I'm trying to do with this project. So the first is just to get the website up and going. So here is a screenshot of what the homepage looks like, and a screenshot to the right of what a molecular entry looks like. So we'll have molecular entries, we'll have entries on reactions, and entries on studies that describes the study. And the plots will be interactive, so you'll be able to zoom in on the plot, hover over, and get the exact mass to charge ratio, for instance, for a mass spectrum. The second objective is to build out the functionality of the database. So in particular, building out plugins that allow proprietary conversion of uploaded data sets. So this is one thing that I think really gets in the way of people sharing their data, is we analyze all of these samples using really sophisticated equipment, but the data comes out in these proprietary formats that you cannot open unless you have the thousands of dollars worth of software, and each instrument has its own software. It's just not possible for everyone to have all of the software. But to convert it to open format also takes a lot of time. A lot of time you have to do it like in the software itself, one spectrum at a time. And so for folks who may have who are sitting on perhaps a lot of mass spec or NMR data, they might be discouraged from sharing just because it takes a long time to convert it to open source, and nobody really has that time. And so I want to be able to build a plugin that will facilitate that process for people, to really lower the barriers to sharing your data. Another thing that I'm aiming to build is a plugin that will allow structure-based queries, so such as what's shown in this figure here with Marvin.js. Like if you ever go to Millipore Sigma and you're looking for a chemical compound, you can draw it and it'll search their database to see if they're selling that compound or something similar. And so I want something like that for the database as well. So you could draw the compound of interest and mark how similar or structure you want to narrow your search to see if that's been reported in the database. And then lastly, applying classifier as well to enable labels for each of the entries for people to use for machine learning. The third objective is actually populating and analyzing the database. So first we'll have alpha testing with our science committee beta testing with the Young Scientist program, and then eventually opening it also up for community to beta test as well. And then lastly, the fourth objective is really working on advertising and getting community buy-in. So we have scheduled 3 workshops for this. The first one just passed, happened in late July, and then we have another 2 for each of the 2 years left of the project. So this workshop that just happened in late July was virtual, and I invited speakers from the metabolomics, chemistry, planetary science, and astrobiology communities to talk about their databases and lessons learned, and really to get the brain juices going for discussion that we had later breakouts groups for. If you're interested, all of the talks have been recorded and they are available online. So if you just type in Astrobiology Spectral Database Workshop and you go to our website, you can click and listen to the recorded talks. So this first year, we started off in January, initiating the development of the database. We had our workshop, and then right now we are working on finalizing the website and getting it ready for people to submit their data. And then starting early next year, we're going to be presenting at OPSC and AdSciCon about lessons learned from the workshop that we had in July, and start opening it up for alpha testing with our science committee in spring, and later beta testing in the summer with the Blue Marble Space in the summer science interns, the Young Scientist program interns. And then in the fall, we will have a workshop that demos how to upload your data, a tutorial, and really open it up for the community to start testing. And then in our third year, we hope to have a workshop at LPSC to once again introduce to folks how they can upload their data, and have a third workshop showcasing what people have done with the data, and we're really focused on getting community feedback, with ending the project on a publication announcing the Astrobiology Spectral Database. So I have 3 conclusions for this talk that I really want to emphasize. The first is that mission work is absolutely critical for moving the origin of life field forward. We need to be able to benchmark what we do in the lab, and what we learn in planetary samples, in situ or return, can really help inform what we can do in the lab, because we don't have all the time in the world to simulate all of the different environments that are possible. And so it's really critical. Unfortunately, ordinal life is not really a high priority for a lot of missions because it's not considered easy to get that science closure. But the potential solution, as I mentioned to you all, is working off of a framework. So instead of just saying, is an ordinal life event possible on here, asking pointed questions such as, can you get certain chemistry if you don't have land, like condensation reactions? And that can be very useful for informing conditions important for origin of life chemistry. The second main problem is, in trying to understand chemical evolution, we need to have a holistic understanding of the organic chemistry, but we tend to just be focused on biologically relevant compounds. And that's because there's just so much compounds that are generated abiotically. We don't have commercial standards. We don't have the time to analyze them. And because of that, a lot of the information that is needed to actually get a holistic understanding is lost. Computational strategies and machine learning can provide an unbiased way to analyze these complex samples. Unfortunately, machine learning is very data hungry. And so because of that, we really need to prioritize the development of a database that is tailored to storing spectral data of abiotic samples, either generated in the lab or abiotic spectra from organic extracts from return samples. And that would be really helpful for building machine learning models that are especially tailored for answering this kind of question. And with that, I thank you all for your time and attention, and I'm happy to answer any questions.
Thank you so much. Laura, do we have any questions from the audience, either online or here in person? Quentin.
I guess I'll go ahead and ask a question. Excuse me. That was really cool, especially seeing like machine learning and how you're like utilizing machine learning in this. So I noticed you use the Spec for machine learning, kind of to try to classify with the spectra. Are there other machine learning programs that you're trying to use like for a similar objective to compare and contrast the Spec and seeing how?
Yeah. Yes, the cool thing about leveraging machine learning is that, especially in the metabolomics community, there are just paper after paper after paper coming out with new models and new ways to analyze data. So already there are several different models that have come out that I'm interested in testing. There's a paper called Canopus. Well, the paper's not called Canopus, but they built a model called Canopus. That's another one that I'm interested in testing. So that's what I'm working on right now, is kind of comparing and contrasting these different models.
Awesome.
Thank you. Chip.
Yeah, Laura, first I just wanted to thank you. This is Chip Shearer. I just wanted to thank you for giving this presentation today. It was terrific. And it's really nice to get some additional insights, what's going on with the astrobiology aspect of planetary science. So this was terrific for all of us. The question I had for you is, there's so much that you're doing right now. Are there any advantages at all for understanding the origin of life that's tied to a particular type of sample return mission? Obviously, that would give you more instrumentation that you could use on these samples and be able to have different groups looking at these samples. So is there any advantage? And then where would you go? What sort of material would you return?
Yeah, really fun questions. Definitely sample return is the best for organic characterization, just because you're with in situ characterization, you're just very limited, unfortunately, and like you can't take a whole laboratory to space. And even then, one laboratory does not have all of the instrumentation that you would need. So sample return is absolutely key, I think, for really moving forward the field. Although there's still a lot we can get with in situ characterization, such as we've learned a lot from Cassini, for instance. In particular, I think it would be great to get samples. I mean, I want more sample return to happen because that would be really exciting. But I think what would really be helpful is just because of, for instance, one of those driving questions that's been asked is land important? What can you get? How far can you get in a bulk ocean where maybe you're limited to the chemistry, to freeze-thaw cycling, or chemistry at the seafloor? I think it would be really interesting to get return samples from ocean worlds like Europa or Enceladus. Fortunately, they're so far that by the time you get them and then bring them back, you're it's probably past my lifetime. So I'm also really interested in Ceres. I think Ceres is another really interesting one because it's a relic ocean world, potentially even has some transient liquid water on it today, and so we could even get at that question with Ceres, and that's not that far away.
Great. And again, in the future, there are National Academy groups looking at ways of, actually doing sample return from the outer solar system in a more timely manner in the order of, in 2 or 3 decades, 5 decades from now. So by the time you're a mid-career to later career person, that capability may be available.
Yeah, definitely.
Okay, we have a hand up from Francis McGovern. Go ahead.
Yeah, hi Laura. As a fellow LPI staff scientist who's interested in machine learning, for my application would be analyzing the shapes of volcanoes on planets like Venus. I was wondering how, the learning curve for getting up to speed on that software, and how long have you been using it, and have you found it easy? Do you need to rely on other folks to help train you, or?
Yeah, that's a great question. I did not take any, I think machine learning started to really take off when I was in grad school. So I didn't actually take any formal courses on machine learning or even coding until I didn't even realize how
Okay.
useful it could be until the end of my PhD. When I was like, oh man, I could have used this. That would have been really useful. So it was my postdoc really that I started self-teaching. So it's definitely, I think it's just, you got to take the time to crash and burn a few times. Just with there's such wonderful resources available online that you can use to I guarantee you run into an issue, you just type it into Google, somebody else has run into this issue, and there's that you can learn from. And so it's a little bit slow, but the more you use it, the easier it gets. And a lot of, there are a lot of fantastic studies like that Spectral. They have their models and tutorials in Jupyter notebooks available that go basically bit by bit. And so a lot of folks are trying to make it easier for people to replicate their data and to use their models. And so I think it's getting easier. I won't say it's like, you can just sit down and immediately take off. So there's a little bit of you have to be a little bit stubborn about it, but it's definitely doable for folks. And I'll take this moment to advertise that we're trying to actually develop a short course on machine learning for sample science that we hope to have next year as part of LPI's Encompass program.
Excellent. Cool. Thanks. I may contact you offline to ask a few more questions.
Yeah.
But great presentation.
Thank you.
Thank you. Anybody else here in the room or any other hands or anything in the chat, feel free to ask there if you'd like. Chip.
Yeah, hey, Laura, I really applaud your effort in trying to coerce your colleagues into making sure, that sort of data is is available. And I know in a lot of the sample studies that were carried out, decades ago, you have to really look really hard to unearth the original data. But recently, there's been, a real move in journals to make sure all of the data is published somewhere. Is that not true in organic studies, such as and trying to put organic databases, that journals aren't requesting some of this data or all of the data being published at some website prior to publication of the paper?
Yeah, it's a really recent development of the push for open science, at least in the organic side of things. And because there is no central repository, if people are going, they are encouraged now by journals, definitely like JGR Plants, for instance, to make their data available. But when they make their data available, sometimes it's not clear to me if journals are actually requiring the raw data to be available. So usually it'll be like data tables of, let's say, the product they identified and the intensity. So what they use to build this particular graphs in the figures that are in the paper, but not necessarily the spectra. And even if they do, because there is no central repository for abiotic chemistry, it's going to be really hard to go and find these data sets that are made available. Like, for instance, if people put it on Zenodo, you have to go and look for these individual ones. There's no easy way to look at this. There is the AHEAD database, which is aiming to try and collate like all of the astrobiology other astrobiology databases that are out there, but it's not designed specifically to facilitate the exploration of raw spectral data. And that's really what I want this database to be, is to enable exploration of the data, export of the data so that you can use it. And so, yeah, I think there's a real need for it.
Okay. Thank you so much, Laura. That's wonderful. And another round of applause.
Thank you all.
Hopefully see everybody next week, same time.